Greetings, ladies and gentlemen, and welcome to the U.S. Transhumanist Party Virtual Enlightenment Salon of April 11th, 2021. My name is Gennady Stoliarov II, and I am the chairman of the United States Transhumanist Party. With us, we have an illustrious panel of U.S. Transhumanist Party officers and advisors. We have our Director of Applied Innovation, David Shoemaker. We have our Director of Visual Art, Art Ramon Garcia. We have our Director of Scholarship, Dan Elton. We have our Foreign Ambassador in Spain and Technology Advisor, Jose Cordero. And of course, we have our 2020 US Presidential Candidate and Director of Marketing, Charlie Kam. And our special guest today is a distinguished individual, author of the book, Replacing Aging, Jean M. Hébert. Is professor in the Dominick P. Purpura Department of Neuroscience and professor in the Department of Genetics at the Albert Einstein College of Medicine. His research is focused on devising methods of cell replacement for the adult neocortex when its cells are lost due to damage or age-related degeneration. And his book, Replacing Aging, really offers quite a fascinating possibility for essentially overcoming the deterioration that the brain experiences over the course of life. It's quite an innovative idea, and we look forward to hearing Jean present it. Jean, please proceed.
Well, first, thanks so much for having me. It's quite the opportunity, and I greatly appreciate it. I look forward to getting some feedback and questions from you guys as well. So I can share my screen now. Okay. And if you can confirm that you see one slide on the screen.
Yes, indeed.
All right, great, we're good to go. So I have this rather bold title of Brain Tissue Replacement as the Cornerstone to Beating Aging. And I will argue, why I think that might be the case. And then, you may or may not agree with me, but it at the very least should foster some interesting discussion in the end. I'm going to start off first with something maybe a little less controversial, and that's something I think we all agree on. We all want to be healthy, and diseases are something that interferes with health, and aging, I think most of us agree, is the biggest predisposition to diseases and being unhealthy. And aging can also directly impacts health without causing disease. So, the frailty that comes with old age, for example, you'd be surprised to find out how many individuals 90 years and older die of just falling down. So, we become very frail as we get older. So even when we're considered healthy and disease-free for that age. So where there's a little more maybe room for discussion and new ideas is how we're going to fight aging or even beat aging. And so what I'm putting forth is this is going to be done most effectively with tissue replacements. And that's great if you can also replace the brain. And obviously not as a whole organ, so I'll go over that in more detail. But if you have a young body without a young brain, that's not very useful. So that's why, you sort of already get the idea of where I'm coming from when I say brain tissue replacement as the cornerstone to beating aging. So I'll start with a fairly straightforward question. What is aging? And I know many of you are very educated in the longevity field, so it may seem odd to ask this question, but I think it's often, the answer is often overlooked. and lost in all the effects of aging that we look at. So if we take a cell, for example, it's in its extracellular matrix. Really what aging is at its core is the accumulation of damage to all the macromolecules that make up this cell and its extracellular matrix over time. Right, so we have proteins, DNA, carbohydrates, lipids, and they all succumb to complex forms of damage. And just to give you, a taste of that, this is the kinds of damage that occurs to proteins alone. So you get cross-linking, you get glycation, oxidation, denaturation, depurination, and all these others. And this is just a subset. And the damage is equally complex to the other classes of macromolecules, the nucleic acids, including DNA, carbohydrates, and lipids. So this complexity is often ignored by longevity researchers, and the damage itself is often ignored by longevity researchers who are looking to oftentimes use pharmacological approaches to sort of either interfering with the accumulation of damage or enhancing the mechanisms that might reduce the rate of damage or one particular type of damage. But those mechanisms are very complex, and they're integrated with the life-sustaining metabolic pathways that occur in all our cells. So if we're trying to, tweak or modify those pathways, we're going to be more than likely interfering with essential life-sustaining processes as well. And I think we already see that in terms of the limitations that drugs have, even for treating simple diseases. It's often hard to come up with a drug where the side effects don't outweigh the benefits, right? And so I think when we're thinking of all these pathways and that are affected with aging, and trying to target them all with pharmaceuticals, I think it's hard for me to imagine how that would occur successfully. And even then, if we were able to tweak these pathways in such a way that we could slow the accumulation of damage, we're still really not addressing the cause, right? The bulk of the damage that occurs. And it would be important to target all the pathways because we know from diseases like Werner's syndrome and Huntington-Gilford that you can get what looks like accelerated aging when even one of these pathways is not functioning correctly. So, I think it's very difficult to imagine how pharmaceuticals will get us there. And, this is described in more detail in my book, if you're interested. And of course, I'd be happy to expand upon it in the discussion as well. So the conclusion that I've drawn from this is that this complexity of damage mandates a replacement approach. So if you can replace a tissue, you remove all the damage at once. And if you're replacing it with a young, pristine tissue, that makes it young again. And so let's look at replacement as an approach. So because I've been focused on the brain, so I haven't paid too much attention to the rest of the body. But when I looked to see what has been replaced in the human body, I was quite surprised to find out that every part of the human body, except the brain, has been surgically replaced in humans. Right? Every internal organ, every limb, every extension part of the body, face, nose, neck, jaw, I mean, you name it, the sex organs, everything's been replaced. The problem is for the biological replacements, those have come from deceased donors in most cases, except for kidneys where a kind person might donate one of theirs. So that's the problem. We can't depend on deceased donors for our replacement parts. But this is just to say that the implementation of replacements has already taken place, right? Where the problem lies is in getting enough replacement parts to treat people for the sake of living longer, healthier lives. And what's going on now in many labs across the world is a big effort to grow organs in the lab, right? So every internal organ is currently in preclinical development. Some have already been transplanted in humans and have been functioning well for quite some time. The bladder is a standout example from Anthony Atala's group at Wake Forest, where entirely lab-grown bladders have been functioning in people now, I think, for 15 years. But there are other examples as well. And like I said, all the other body parts are being worked on. And of course, there's also replacements with prosthetics. So the technology there is improving constantly to the point where the artificial limbs now are very close in terms of performance to natural limbs, and they're just going to improve as well as artificial hearts have been functioning in people for a long time. In fact, there's an example of a particular heart valve pump that, after an elderly person dies from other causes, that pump will still be functioning in them. So blood is still circulating. So that's kind of weird, but it just goes to show you that the technology has improved to that point. So these replacements are going to happen for the body. That's sort of inevitable. And but we can't replace the brain, at least not as a whole. But everything we know about how the brain works tells us that it should be replaceable progressively over time, right? And so that's sort of the central point of the work that we're doing, and that I'm going to share a little more in detail with you now. But you may first ask, how is that possible? Why can you change even progressively your brain over time without changing who you are? And the answer is yes. And that's because the brain, and particularly the neocortex, so that's the outer part of the brain that's all folded, that we think of when we think of a picture of the human brain is the neocortex, this outer part. And it's remarkably plastic. Our memories and thought patterns are not statically recorded in a particular set of neurons or synapses. Things are much more dynamic than that, and they change over time. And that's why we're able to learn new things. So the brain already is, normally changes over time, the neocortex in particular. And that's why we forget things as well, because we're not, it's not an accurate recording device. It's a live, living tissue. And we've known for a long time that if part of the neocortex doesn't get used, it will get taken over by other functions. But my favorite example of plasticity is in humans of a fairly advanced age that have a benign glioma that had been growing in their language center for 5 to 8 years, that completely has eaten away their original language center. And during that time, these individuals have not lost their ability to speak. And there's no change in their personality. No one notices any difference in them, the people that are closest to them, their family and friends. And even after they remove the tumor with surrounding tissue for good measure, in most cases, they can still speak perfectly fine, right? And so this group in France actually, was quite surprised to find that this was the case. So they started studying this and mapping where the language center was in these patients. And they they realized with awake brain electrode mapping and functional MRI that the language center had moved to a new place in the neocortex. And they knew for sure it had moved because they had a low number of patients, but some that had a recurrence of the tumor growth over the course of another few years. And so they had to go in again, remove the tumor, and map where the language center was now, and it was now in a new place. So they had a record of movement of language. And so if something as dear to us and as complex as language can move from one neural substrate to another over time, this tells us that in principle, we should be able to replace the neocortex progressively over time without losing our sense of self and our abilities, such as language and our memories, as long as we keep using them. So that's one of the reasons. The other reason why it makes sense to think that we can rejuvenate the brain progressively over time with cell replacements or tissue replacements is a more recent findings over the last 5 to 7 years, showing that if you transplant immature neocortical precursor cells in the adult neocortex, and this is in animal models now, there's less evidence of this in humans so far, but if you transplant these immature neocortical precursors, they'll differentiate as they would if they were in the fetus and postnatally in the early years of development. So they'll project to their normal targets and they'll form synapses with those targets. They'll receive the appropriate input, and to some, as far as people have looked, they seem to function normally. So that also tells us why replacements should be possible using these immature cells, and if done progressively over time, to allow plasticity to have functions move to these new substrates. And I'll show you a little bit of data from our lab. Very little data in this talk. It's mostly the general ideas, but I'll show you a little bit about what we're doing as well. So I'll show you some of that as well. But this is the sort of the overview of the process. So the neocortex is not just comprised of neurons, but several cell types outlined here. I guess you can see my cursor. And these cell types can be patient-derived, as many of us have heard of before, I hope. So you can take a skin biopsy, for example, collect the fibroblasts, and those can be reprogrammed into what are essentially embryonic stem cells, or what are called induced pluripotent stem cells. And these, or embryonic stem cells, are sources of cells, stem cells, that can give rise to basically every cell type in the body. And protocols already exist for generating the different cell types that make up the neocortex. So all this has already been done with human cells. What hasn't been done is what we're working on, and that's reconstructing or reassembling the tissue from these cell types in the aged or damaged neocortex. Right? So that's what we're aiming to do. And our first clinical case study will not be aging, because that wouldn't be practical and be very difficult to get approved at first. It will be something like stroke. In fact, stroke is one of the leading causes of brain damage, and it leaves patients generally disabled for the rest of their lives. There's some compensation that occurs, again, because of plasticity, but this damage occurred, unlike the benign glioma that took 5 to 8 years, a stroke occurs in seconds, and the damage, occurs within, hours or maybe a day. It continues to occur, but it's a very catastrophic event. So there's no time for functions to move to a new substrate. But providing a new substrate should make it easy for the patient to relearn the lost functions. So the idea is just you get the stroke, you remove the dead tissue, the damaged tissue is not actually dead, but it's filled with glia instead of neurons. You remove the sort of the useless tissue, and then we use our approach to rebuild functional tissue here. So the neocortex is a multi-layered structure. It has different layers of neurons. And so that's part of our approach, is to layer in the deep precursors, then the mid ones, and then the upper ones on top of them to recreate the normal cytoarchitecture of the neocortex so that it's functional. And there's a little, I'm making it sound pretty simple here, and there's more to it than that, and maybe we can get into that in the discussion. But that's essentially, what the approach looks like. And for aging, it's very similar. We do the same engraftment, except we do it in sort of a few cycles. So here's a young brain on the left. As we get old, the brain atrophies, which is not good, but the silver lining is it creates space for initial grafts without having to remove any of the old tissue. So we can put in these young grafts, sort of distributed in the old neocortex, and then they take a while to differentiate, up to a year, and then to integrate and adopt functions. It takes 3 to 5 years, and during that time, as these new substrates are being adopted by the brain, and we begin to use them, at the same time, what we'll be doing is slowly silencing other parts of the neocortex, just like the benign glioma did in the example I gave previously, but without using a glioma. So we would do this with other means, pharmacological, or there's even some physical means that might work to progressively silence parts of the neocortex so that those functions are forced to leave that tissue that's becoming silenced and move to other parts of the neocortex, including these young differentiating transplant-derived areas. And then once these parts that we're silencing are completely silenced, then they're useless. Right? So they are removed and again replaced with young immature tissue that then differentiates, integrates, and adopts functions. And we basically repeat this for a few cycles until the entire neocortex is rejuvenated. I'm excluding the other parts of the brain here, but they would also need to be rejuvenated in a similar way, and coincidentally with these replacements not in a random way, but where the functions for the different parts of the brain, for example, the auditory nuclei in subcortical parts of the brain are developing at the same time as the auditory cortex, right? So they will integrate much more easily with each other. Again, this is an overview. There are many more details to this that I'm leaving out, but that's essentially the long-term plan. Long-term, if optimistically we start doing this in 10 years from now, but it might be a bit longer than that. So this is the little bit of data from my lab that I'm showing you. We have a lot more than this, but we use the adult mouse neocortex as a platform for rebuilding the human neocortex. And so here we make these canonical lesions. We insert our mixed cell populations to fill the lesion. And these cells have both neuronal precursors, neocortical precursors, and as others have shown as well, as I mentioned earlier, these neurons differentiate, send projections to appropriate targets like the corpus callosum and the striatum, where they synaptically integrate. And we've shown this, as has other groups, with both mouse cells and human cells. But we're really focused on trying to develop human neocortical tissue in situ. And the idea here is not that we're going to make it in mouse and then transplant it in human. The idea is just to figure out how to make it in mouse first, and then make it the same way at the site of the lesion in humans. So we also have vascular cells that we include in our grafts, and they come together and form blood vessels very quickly, and they fuse with the host vasculature, and they circulate blood. You can see the little green dots moving really fast. Those are just fluorescently labeled red blood cells to make it easy for us to see flow. So both the neurons and the vascular cells integrate with the host in our graft. So that's really good, but it's really just a starting point, right? So, here we've added one class of neurons with the vascular cells, but there are, as I mentioned earlier, these other cell types that are essential to the neocortical development and function. And so we need to include those as well. And as I showed in the initial outline slide, all of those cell types can be derived from human pluripotent stem cells. So that's good. We just haven't done it yet. And so that's our first major aim, is to reassemble these cell types. And as I've described already, it's to reassemble them, the precursors, in such a way that they differentiate in these appropriate layers, which are necessary for optimal function. And then another aim that we're working on in parallel is to show proof of concept that that neocortical tissue replacement can work. I think that's an important point. And so here's just an outline of that experiment. I don't want to spend too much time on it. And then I think this is my last slide, so I think we're okay on time. But essentially, the way we're testing this is we take a recipient individual, and we train this individual, not a human, it's an animal, to perform a task that is fairly complicated. So it requires multisensory integration of a sound and a particular visual cue. And then the recipient then needs to push a lever or pull a trigger to get a reward. And they learn this task. It's like a game. They get very, very good at it. And then what we do is remove just the part of the neocortex that controls the output of that limb to press the lever or pull the trigger. And we replace it with our immature tissue. And then we can confirm after surgery that the tissue, although the recipient still knows how to play the game, they cannot execute it because the last link in execution is the motor cortex. Well, apart from the spinal cord and the actual limb, but in the brain, it's the motor cortex. And so they want to play, but they can't. So then we monitor for the next few months the differentiation of our graft to know when the neurons are coming online and becoming active. Once that's happening, we reinitiate the behavioral training so that the animal then plays the game again. And the expectation is that that the new tissue will adopt this function and be able to allow the animal to execute the task. And then we can prove this by silencing specifically the neurons that we put in without affecting all the other neurons in the recipient using this trick, this chemogenetic trick that we have. And I can explain that later if people are interested. But it's a way of just silencing our transplanted neurons transiently. So it's a chemical induced silencing, and once that chemical washes out of the body, then the recipient should be able to play the game again. And proving that our graft actually has replaced the previous tissue that was there in a functional, useful way to the animal. So that will be important. So those are our two aims, and that's pretty much all I have to say. This is a big project with many moving parts, many areas of expertise. So we're not doing this alone, and we can't do it alone, especially moving forward. So we've started collaborations with all these other labs that are excited about this project. Of course, it costs money, and that's a whole other question. So we, that we're working on is to get sort of this concerted funding for this project to get all of us working on it in a much more in a way that accelerates progress a lot more. Right? So I think that's it. Yeah. So just to recap the main thesis. So in combination with replacements for the other parts of the body, that's why I think success in brain tissue replacement will mean that we can actually defeat aging and not just, incrementally slow it down, with some pharmacological approaches. So, you can let me know if you agree with that or not, or what your thoughts are, criticisms, comments.
Wonderful. Thank you, Jean. We appreciate this presentation. It was quite informative for us. And now let us go to Jose Cordero for his question.
Oops.
Well, first of all, congratulations. This book was fantastic. It was my Christmas reading, and I truly thought this was revolutionary. I have been following, obviously, longevity, anti-aging, but this book has a totally different perspective, completely new to me. So I was fascinated. So I truly congratulate you, and I am happy to see that your effort is being followed and shared with other researchers. So I basically have like two questions. Well, first of all, not only did I read your fantastic book, but I also read some other books that you obviously know as well that just came out about the same time, Ageless, with Andrew Steele from the UK, and your colleague at Einstein College near Barcelona, Age Later. But these two books, in a way, are relatively normal in the anti-aging longevity field. Similarly with David Sinclair and Lifespan. All those books, which are incredible, fascinating, and I am so happy they just came out in about 1 year. They are nonetheless normal for people who follow these. But your book truly is revolutionary. So I want to know what else do you see coming in your area, and how does it compare, and how do you collaborate with people in the more traditional longevity realm? And one final point, because your book actually, in terms of sales, I was checking on Amazon.com, and it is relatively low compared to the other ones. And why is that? Because it looks like self-publishing maybe, or at a smaller publishing house. Because I want to help. I mean, that book is fantastic, revolutionary. So it has to be a major bestseller throughout the world. So thank you, and I hope to hear from you.
Yeah, well, thanks so much. That's very kind of you to say. Yeah, it was a small publishing house. It's actually somebody you might know, Jan Vij, who's in the longevity field, the aging field. He studies aging. And he has a publishing house that's rather small, but he was very, it was great working with him because we both liked the ideas. There was a very good back and forth that you wouldn't get with a bigger publishing house. But yeah, we don't necessarily get the promotion you get with big publishing houses. But yeah, I should probably hire you to promote it. Thank you.
All right.
Okay, but the other researchers working with you, and how do you see this sector moving?
Yeah, so right now, I hope they come together, right? I think collaboration is important. There definitely can be areas of synergism, like David Sinclair has talked a lot about epigenetics. There's obviously limitations to how that can be applied, but there's also benefits that can come in combination with replacing tissues, where you can allow the host environment perhaps to be a little more regenerative and accommodating to grafted tissue. That's just one example, right? But so I think it is important to work together, even though in the book I sort of present the two approaches as very different. But there is obviously room for overlap and working together. And so I think that's important moving forward. Right now, they're quite distinct, though. You're saying it's revolutionary because the longevity field does not acknowledge replacements as a viable approach to defeating aging. And I think it's going to be quite the opposite. I think it's going to be a very impactful approach. And so, yeah, so the idea now is to have it move forward faster and get more people excited about it, invested in it, and working on it.
Yeah, I have some questions. Yeah, I was really curious why exactly the brain shrinks with aging. Is that because of actual loss of neurons, or is it just deterioration?
Yeah, so you would think it's something that should be known and pretty straightforward, right? But it's actually been difficult. It's difficult to quantify number of neurons in a brain. And the numbers have changed over the years. For example, we've all heard we only use 10% of our brain, and that's a complete misrepresentation of where that came from, which is basically that most of the cells in our brains are not neurons, right? Only 10% are neurons. But that number has changed over the years and has gone as high as 45% of our brains are neurons, and the other 55% are non-neuronal cells, like support cells, all the glia that are essential to the functioning of these neurons, the vascular cells. Those are all non-neuronal cells, right? So it's hard to get accurate quantification, and there's still debate, even in the normal brain, of how many neurons you have. And so it's hard to compare in an old brain just how much loss of neurons there are. And we've sort of heard that both, that there is some loss of neurons, or it's negligible, or there's very little loss of neurons. What is not controversial, though, is the loss of the complexity of the projections from the neurons. So that is clear that shrinks. If you look at old neurons versus young neurons, the young neurons have these very elaborate dendritic trees with axons and lots of branches. The old neurons they look old. they have much more restricted branching and they just don't have as much to communicate with.
Well, I just want to jump in and ask you another question on that point. If you graft in this new tissue do you expect it will remain young and plastic, or will it be sort of contaminated by the aging environment around it, so to speak?
Yeah. No, that's a very good question, and one that we'll have to keep an eye on. The data so far is that if the young tissue can stay young and functional for quite some time. The data, there's a growing amount of data in animal evidence that suggests that that's the case, that it can function totally fine, even in an environment where there's inflammation.
Yeah.
In humans, there's much less evidence, but the evidence comes from the transplants for Parkinson's disease, where even though the environment is not great, the neurons that were transplanted look like they function well for at least a decade and a half without succumbing to that same degeneration. Right. So it's something that we'll have to keep in mind, and of course, it depends too to how much we want to change the parts of the brain at once. So the quicker we rejuvenate, the less this is an issue. But the downside of that is you don't want to lose who you are. You don't want to revert to being an infant, learning everything for the first time. You want to maintain your memories and your self-identity. So
I just want to ask you a few other questions. So is it actually the case that new space would open up in the skull for you to put in these grafts, or would you have to remove old tissue? Because that's kind of what you showed was putting these grafts of tissue in. And I was thinking, like, if you're actually adding more neocortex, that's almost like an enhancement, I know later the theory is the kind of plan right now would be you would kind of swap out, but at least when you first put that new tissue in, that almost seems like an enhancement. Would you agree with that?
It is an enhancement. It provides the substrate for enhancement, and that's the whole idea. So the way that the brain of an older person works is very different than the way that the neocortex of a younger person works, and this is shown by functional MRI in humans, where a young person, everything is really well compartmentalized and efficiently used. An old person, the functions are much more diffuse and widespread. And that's because of the degeneration of the neurons, not necessarily the loss of neuron numbers, but the loss of connection. So it takes more neurons doing this, participating in the same task to get it done. So this is plasticity at work, even in the old degenerating brain. So that brain is just dying for new substrate to work with. And so if you're putting in these young substrates, it's gonna enhance function, and that's the idea.
Yeah, I just want to have one last question, which I'm very curious about, which is how dangerous is it now to actually breach the skull? And this seems like a very relevant question here, and also for brain-computer interfaces. Just how risky is that? Like, what are the risks of infection or something like that during the surgery?
Sure. I mean, that's always a concern. But to put things in perspective, people are having brain surgery like routinely, right? All around the world every day. Some of these are major surgeries going deep into the brain. Tumors grow in every part of the brain and have to be extracted from every part of the brain. So these surgeries, luckily, there's some very good surgeons out there who can, with minimal damage, go in and remove things, so presumably add things as well. So, having said that, though, as we get older, the chances of getting, secondary effects or bad consequences from the surgery, like bleeding, infections, or clots, increases. So that's something that needs to be taken into account as well. So the earlier we can start doing this, the better. You don't want to wait till you're like 90 necessarily, because your resilience is much less, and so your ability to or your risk of succumbing to the bad consequences of surgery are greater.
Yeah, I have some other questions I'd love to pick your brain about, but I think maybe we should let people from the audience get their questions answered at this point.
Yes, so we do have some audience questions that I think will be helpful for advancing this discussion. Daniel Twett asks, couldn't brain function be enhanced without the need for progressive silencing? Has that option been studied? And of course, in your first case where you discuss the stroke victims, the silencing has been done already, essentially, because of the damage from the stroke. So you can take out that damaged portion and then replace it with a new graft. But do you have to silence healthy tissue, or can you just graft on new tissue, and perhaps as the old tissue dies on its own, you remove it at that time, and that frees up the space for yet additional grafts?
Yeah, I mean, that's an interesting question because in theory, right, you shouldn't have to remove the old tissue as long as you're providing young new substrate that will enhance function. But unless, there's a space limitation if we want to keep the neocortex and skull, within our skull, the size of our head, the current size of our head. And it's hard to imagine how you might go well beyond that and still maintain normal connectivity. So I think we do want to stay within the context of our current head and brain and skull sizes. And so, I think in that case, if you really do want to rejuvenate the brain, you do have to replace all the parts. You don't want a really old part where an aneurysm or a stroke is going to increasingly be likely to occur and going to affect your young tissue as well, right? You want to proactively, I think, take the steps necessary to rejuvenate it. But you're right, in theory, adding new tissue by itself should be highly beneficial, but I think we also want to get rid of the old tissue.
And this raises another interesting set of questions if you do get rid of older tissues. T. Theodorus Ibrahim asks, how much neocortex could you replace without losing identity or memories? And I understand the argument that essentially over time, other areas of the brain will take over the functions of the previous areas. However, there does have to be a certain gradual migration there. So how much would be too much in terms of removing tissue that would hinder the functioning of the brain or cause a person to forget some essential component about their identity or past experience of life?
Yeah, another really good question, and to which there is no definitive answer yet. Some of this will have to be determined empirically. From our stroke trials, we'll get some information about how long it took for that new tissue to adopt function, for example. So as we go, we're going to learn more about this process. There's going to be obviously a balance, right? Just like Janady explained, it's too much silencing too quickly, you're going to forget things without giving time for those functions to transfer to new substrate. So the amount and the timing of replacement is something that we can take a good guess at now, but we'll have to very closely monitor and adjust as we develop this approach.
So in terms of the amount and kind of testing that has been performed already, Didier Kernel asks, was something already tested on animals? And he points out that brains are probably the organs that vary the most between animals and humans. So a follow-up on that would be, to what extent are you confident that results which are manifested in animals such as mice will be upheld in subsequent human testing?
Yeah, so a very important question, and not enough is known about how these grafts would perform in mammals. And I say mammals because mammals are the only class of animals that have a neocortex. In mammals that have bigger, more complex neocortices, right? They have sulci and gyri, so those folds and dips. And so that's, in our 5-year plan is what we intend to look at, because that is very important to show that the connectivity that is regained even with human cells, but in a mouse host, that can also occur in larger brains. So yeah, we're looking into doing that as one of the as soon as we can. Very important one.
Yes, indeed. So David Wood asks, as well as replacing cells in the neocortex, what about cells in the underlying brainstem? Could or should they be replaced as well?
Yeah, I mean, I very briefly alluded to the fact that I'm focusing on the neocortex, but every part of the brain needs to be considered and replaced appropriately. And the brainstem, obviously, without a brainstem, we stop breathing, we essentially die, right? So we can't neglect the brainstem or components of the midbrain, the thalamus, for example. Very important. And the idea is that after we've shown that this works for stroke, during that time, we'll also be developing the cell populations necessary to replace those other parts of the brain in a similar way, so that these transplants, again, as I very briefly alluded to, would be done sort of coincidentally. So for certain functions, right, like the hearing, you would do it unilaterally for starters, right, because then you could always use the other side functioning well. But you would not just replace the auditory portion of the neocortex, but the auditory portion of the thalamus, or at least half of it, as well as other parts that are not in the brainstem, but close to the brainstem in the hindbrain area that are also important for hearing, for example. So those would be replaced in a coordinated way throughout the brain.
So one of our viewers with a fitting name, Why Not Now, asks, when will these treatments be available?
Yeah, I mean, I think optimistically, in 10 years, we could start trials for things like senility or dementia, which are essentially very close to aging. But in terms of full brain age reversal, I think that will take a little longer. Add another 5 to 10 years to that. But again, these things, particularly for this approach, this is an approach to implement that would take in itself 15 years, but you get benefits, right away. So, it's not like this. It's not like the process has to be complete before you're getting the benefits. But yeah, the sooner we can get to that point, the better.
And it's interesting because recently, Aubrey de Grey, who is of course well known for his SENS approach to rejuvenation biotechnology, advanced his time frame for the arrival of longevity escape velocity, essentially stating he believes now there's a 50% probability of reaching it in 15 years by 2036. So the time frame you mentioned dovetails with his time frame as well. And this is a good occasion to bring in another comment by David Wood, who says, quote, "I'm not entirely convinced that Jean's approach is somehow completely different from that proposed by, for example, Andrew Steele and Aubrey de Grey. Andrew Steele recommends that research is continued into each of the 4 types of approach to damage, which are remove, replace, repair, and reprogram. So replace is definitely part of the set of therapies that these other scientists advocating for advances in human longevity are proposing to be researched. So what do you think of that comment? Do you think your approach already has a place within frameworks like SENS or the framework that Andrew Steele advances? And if not, where do you think those frameworks fall short?
Yeah, no, it definitely fits in. And I should say up front that, you know, SENS funds a good part of our research. So, you know, I think we're very much overlapping in the way we see things. And you know, the choice of words, remove, repair, replace. So our approach does all that. Reprogram, you know, you could argue that as well. So yeah, I think there's a big overlap. I think the, you know, Aubre's view is a little more broader than mine in terms of the different approaches and combining them. You know, but he of course, that's what he does. You know, he sees the whole field. He's engaged in everything. You know, we're very focused on our task, which is to be able to achieve brain age reversal. So, you know, I'm maybe not as engaged as he is in all the other tasks, but what we're doing certainly fits in to his view of the world, for sure.
Yes, indeed. And we actually had a virtual Enlightenment Salon with Aubrey in February of this year. Quite a fascinating conversation as well. So hopefully these venues can help provide our viewers with an integrated understanding of ongoing developments in regenerative medicine and rejuvenation biotechnology, and lead people to understand some of the complementarities among the research paths that are being proposed. So now let us go to another question from Mike Lazine, who asks, why can't we find a way for the human body to grow limbs or appendages like fingers and toes naturally, like a number of reptiles do? It seems that there are organisms that have regenerative capabilities that we do not.
Yeah, even for parts of the brain, some of these species will regrow parts of their brain, including the axolotl, I think. So yeah, why can't we? That's, you know, that's obviously an area of intense study and a very good question that a lot of people are trying to answer. With the answer, the hope is that we'll come up with a way of doing it in us. And you know, so far we haven't been able to, but that doesn't mean that we won't be able to. So particularly for things like limbs, right? I mean, that's exactly what we would want to use it for, unless we're happy with the artificial limbs. But again, those artificial limbs, they're going to surpass our natural limbs in not very long. So the question might be moot in terms of limbs, but I mean, it's still, maybe nothing will ever beat the natural limbs. So maybe, yeah, it is worth trying to figure that out. And I think that's an area where, you know, epigenetics can be very useful, because in that case, if your limb is gone, it's essentially replacement. That's the same thing we're proposing. But you're, instead of growing something in the lab, you're growing it in situ, which is actually what we're doing for the neocortex. So, you know, it's very similar. except there we use exogenous cells, and what you're referring to in the axolotl, it uses its own cells. Either way is fine because it's going to be a new, pretty much damage-free tissue, except for the occasional DNA mutations that are not fixable because they're information content, so it's not obvious how they would be fixed, but nevertheless, you would have an essentially young new limb. So, I mean, hopefully somebody figures it out as one potential approach.
Yes, absolutely. And it is quite an interesting question to consider which area will advance more rapidly, the ability to essentially biologically regenerate limbs or just replace them with artificial prosthetics. And one of our viewers, Jennifer Hughes, states in her view, she's pretty sure that with the improvements in regenerative medicine, we will only need prosthetics if people want them, because she thinks with many of the advances in stem cell research, we will be able to repair organs without replacement. So what do you think of that comment? Do you agree with it, or do you think the more mechanical prosthetics will predominate?
Yeah, I don't know. I mean, I was just in Central Park here in New York a few months ago, and I was watching a man run upstairs, and he had one leg was completely, one of these prosthetics, and his motion, his gait was totally natural, and I was stunned. It's like, wow, can I have one of those? I mean, it's very, very natural, and when it gets old, you just replace it with a new one. By then, they'll be even better. So I don't know, and they're improving these prosthetics in the sense that you get feedback from them about feeling, proprioception, which helps your brain embody that limb as part of you, right? So those are new additions that are being made. But the point is well taken. Again, maybe there's no need. I think the prosthetics are leading right now in terms of limb replacements. In terms of internal organs, though, prosthetics are very difficult. The heart is a mechanical organ, so we've had prosthetic hearts for quite some time now, and they're also improving, but they're still not as good as young natural hearts, right? Because they still have complications in terms of the connections and occasional blood clots. So they're still not as good. So maybe, certainly for the other internal organs, those are going to be, the biological approach is leading right now, I would say. And then in terms of regrowing them from cells in situ, again, that's not impossible, but if you want to get rid of the old damaged tissue, you're going to have to remove something. And I think right now it seems to be easier to regrow things in the lab rather than for those internal organs than to regrow them inside of people, because in the meantime they still need something functioning as well. So in terms of that, I think the lab-grown organs are leading the way for the most internal organs, including liver, kidney. The bladder's already there. The intestines are, there's been some recent publications of lab-grown intestines. There's a lot of other examples as well in development.
Mm-hmm. Yes, that's fascinating, and it does seem progress is being made. Different approaches may prevail, at least in the short term, for different areas. So it seems that the lab-grown approach is prevailing for organs, but perhaps the artificial prosthetics are prevailing for limbs, at least right now. And yes, we've seen images, videos of individuals with artificial legs who can even run faster than runners with biological legs. So that is quite fascinating and certainly quite promising. Now, one area that Didier Kernel notes where progress is desperately slow is with regard to neurodegenerative diseases, particularly Alzheimer's disease. And he is curious to know, have you had any contacts with the Alzheimer's research communities, and do you think this approach that you're proposing could benefit patients of Alzheimer's disease as well?
Yes, I do think that it would benefit and even reverse degeneration. Not as confident as with aging. Aging is slower and more progressive. Alzheimer's is like this accelerated degeneration, which makes it a lot more challenging to address. But I think, right now there's nothing that can be done to effectively treat Alzheimer's. So this is an approach that I think has a high potential of working. And so we have, tried to get interest from funding agencies who are focused on Alzheimer's, and they're sticking with drugs. So they're not interested in this. So it's a bit of a, it hasn't happened yet. I think that in time they will adopt this as part of their arsenal. But it's really dominated now by molecule and drug development. But again, I don't have high hopes for those approaches having big impacts, maybe tiny measurable ones at best. But yeah.
So what do you think is the reason why there is such a heavy focus, at least in mainstream medical research, on the drug development approach, especially because pharmaceutical development is exceptionally expensive because of the various FDA requirements? It takes billions of dollars and 10 to 15 years to bring a typical drug to market. And it does seem like a surgical approach, at least in individual cases, would be less expensive, so better capable of being implemented in a decentralized fashion.
Yeah, I mean, I haven't looked into the cost comparisons in detail. One might have naively thought that surgeries in general are more expensive than taking pills, at least at the implementation and the development. And yeah, they may be very similar in terms of cost, because I'm sure the FDA would want to see all sorts of safety measures as well, as they do for current cell transplantation. So that might be a little more similar. But the love affair with drugs has a long history, and there are others who have written about this in more detail, much better than I can explain here. But there's a history of why and reasons why they're so pervasive in our approaches to medicine, often at the exclusion of other potentially effective approaches. But again, I think that's going to change over time.
So David, you have a question. Please feel free to pose it.
Yes, I guess this would be changing gears just a little bit. My question has to do with cryonics, actually. And I'm just wondering if there's a role for this progressive tissue neuron replacement, either in cryonics preservation or cryonics restoration. Have you given that any thought?
So by cryonics restoration, you mean coming out of a cryonic stasis? Or
correct. Okay. Yes.
Did you have any idea of how they connected? I'm not, because basically my understanding of cryonics is you're on pause. Nothing's going on, right? You're sort of
sure.
Not necessarily. I don't think you can use frozen necessarily if you don't have ice crystals, but you're
the vitrification
metabolically in stasis.
The vitrification process maintains the brain structure.
Vitrification, yeah.
And so in my mind, I was thinking that, especially coming out of the cryonics pause process, trying to piece together a way of taking that brain structure and taking brain tissue and somehow melding them together or paralleling them in such a way as to be able to take the information out of the brain structure and put it into that brain tissue. So that's kind of where my mind is going with this.
Yeah. I mean, that's a whole other, there's this whole other field as well where, they want to move or copy your brain to, a synthetic substrates and stuff. And I think that's a discussion. It's a different approach altogether. Whether that maintains who you are or not, or it just makes a copy of you, gets a little complicated and often leads to rather heated discussions. So, that's a sort of outside of what we're doing. But I guess, in principle, just to answer your question, I think if you could, sort of image this vitrified part of the brain in such a way that you can get enough information, whether you could recreate that in naive tissue. I don't know how you would be able to do that because these tissues are very dependent on their environment to differentiate and form normal connections. So yeah, I'm not really seeing much synergism with the sort of the reanimation process and this tissue, this new tissue.
Okay, well, thank you.
Gennady, I had a question for Gene.
Yes, please.
I saw in a previous, I think it was the Foresight interview that you did a couple of weeks ago, and they had some really good questions. One of them being the funding. You suggested, and I know that's really probably the biggest holdback you have right now, right? It's just you need the money, and your suggestion was somewhere, you said between 10 million and 50 million to get things to where they need to be of benefit to all of us. So a couple of questions. One of them is a lot different than that, but one of the other things I heard in that interview was that you had presented this to the Undoing conference in Germany a couple of years ago. Is that mine, right? Okay, so that I'm guessing then you that was presented in front of Michael Grieve and Frank Schuler of Kazoo and Forever Healthy people, right? I mean, and you mentioned a little while ago that you get some funding from SENS, and I know that they fund SENS as well. So I'm wondering, how is it possible? Because what you're doing is fascinating. I got to tell you, I think you're totally on the right track. I think it's in perfect congruence with what's going on with Aubrey. I think, there's a lot of people out there doing body part replacements. Martine Rothblatt and George Church are working on Revivicor and things like that. And most recently, there was an article in that I just saw. In fact, I posted it on Facebook about what they've done in Buffalo, New York, with 3D printing, where they can do it really rapidly now, so that there's no problem with the organs to be viable. But I guess my question is, when you were there in Germany and you presented it to those billionaires, the guys that have all that money, how is it they did not fund you? I mean, what do you think's going on here? Because it sounds like what you're doing is a great thing, and it really needs to be funded. So why is it that they didn't? What do you think's the holdback? How do we convince them? And I want to help you do it. So what are your suggestions? Is it just marketing? Is it a pitch? Is it what? What do you think?
So I think, when I did present at the Undoing Aging, and I presented there, a couple of years ago and a couple of years before that, the work has changed. Our work in the lab and what we found and our vision for how this is going to work has progressed. So at that time, I talked about several approaches that we're taking in the lab. So it wasn't focused just on this, tissue engineering in situ with progressive replacement. We were still looking at other approaches, and we have one other project that's also still ongoing, where we could replace neurons using a cell type that disperses throughout the brain. These are microglia, and so we still have that project going, but I presented that as well. So I think the idea was quite diluted in my presentations, whereas now, I'm emphasizing and focusing specifically on this approach because I think it's the one that will work the best for reversing brain age, whereas some of our other approaches will help maybe, and even treat Alzheimer's, but they won't reverse brain aging. So I think that's why maybe I didn't get much feedback at the time. So since then, and in the last 9 months is when I've really started reaching out to potential funders, and have been in communication with a lot of them for some time now, and they're not necessarily these billionaires though, but they're maybe lower level investors, and so we're trying to build a group that when they come together will have sufficient funding. But I'm not at all against having a billionaire step up to the plate and getting this rolling sooner rather than later. So if you want to put me in touch with any of them, that would be great.
Oh yeah, definitely. So you're open to the idea of having, investors and turning this into something where you can, I think you're right. I mean, if you focus it in a way that someone sees some sort of gain from it all, that's what a lot of investors want, obviously. And it sounds like you've refined your presentation. So that, that could work. I mean, let's stay in touch, and see what I can do to help you.
Excellent. So we have an interesting question about the mechanics of the replacement that you're proposing. This is from our viewer Upgrade 9000Q, who asks, if you want to eventually replace the whole brain, would you need to remove the top part of the skull, or do you need a bunch of holes? Could you do one hole where injected cells travel throughout the brain? And of course, presumably the less, let's say, drilling that one does, the less damage to the skull that one does, the more effective, the safer the procedure would be. So how do you envision that happening?
I think it's ambitious to think that we could do it through just one hole. And maybe one day we can imagine how we might be able to do that. But no, what brain surgeons do now, in many cases, is they cut a window in the skull, and they have the skull flap. It sounds terrible, but then they put it back, it heals, and everything's normal again. So it's I think that's the approach that's going to be used in this case as well, is because you're going to want to access several parts of the brain at the same time, which you can do with a reasonably sized window. So I mean, it sounds unappealing, but it's done regularly already, and so I don't foresee that as being a problem. In fact, there are some people that don't have skulls; they have plates there because of an accident or something. So that can already be replaced. Not that that's ideal though, but and that's also not to say that the surgical technology won't improve with time. I think certainly with AI-assisted robotics, we're already seeing that for certain surgeries where you can do as well as the best surgeons, if not better, because you can do a better job of avoiding any significantly sized blood vessels, for example, and really minimize the damage even when you're going deeper in the brain. The imaging technology as well is getting better, so you can get much better at the surgery. So I think between now and 15 years, I think that surgical techniques are going to improve significantly, which will facilitate things. Maybe not to the point where a single hole will be sufficient to do the surgery, but who knows, maybe one day. But yeah.
Mm-hmm. And certainly improvements in surgical techniques are developments that we should all look forward to. I do want to note, though, that for most people, going in for brain surgery today is quite a rare occurrence, one would hope, and many people do manage to spend their entire lives without needing to do that. So my question to follow up on this is, how often do you envision people needing to undergo this procedure, given that brain aging happens continuously in order to graft on the new tissue? It would be a fairly involved type of undertaking. So once every 10 years, 20 years, or more frequently than that?
I think less frequently. In terms of the, it's going to be more than one surgery within like 2 decades time. You're going to have multiple surgeries, but after that, I think you'll be able to go as long as you go now without surgery. So at least 60, 70, maybe 80 years. But there will be a time when you're going to be maybe a little unhappy about all the surgery and all the follow-ups that go with these surgeries. But because they'll, like I described, there will be cycles of grafting that will go on, over the course of 1 or 2 decades. So yeah, but once your brain is sufficiently rejuvenated back to a younger state, then it should be good for a long time.
Mm-hmm. So one of our viewers, Peace March, suggests that nanotechnology might be incorporated into this kind of replacement. And I'm curious about this too. Do you envision a time when perhaps instead of needing to open up the brain, one could, for instance, take a pill that contains nanorobots and they somehow manage to cross the blood-brain barrier, go into the brain, do the replacements that they need to do, and maybe even stay there and do some periodic repair work on other brain cells. And that would be a kind of routine, non-invasive procedure that people undergo. Do you think that's conceivable? And if so, how far away are we from that kind of approach?
Yeah, I mean, it's very hard, again, to imagine, just like it's hard to imagine how drugs could deal with the complexity of damage or the complexity of the protective and repair mechanisms, because they are so complex and integrated with living tissue and part of the living metabolic pathways. So having nanobots do that, I think, is also hard to imagine. But the more physical role that these nanorobots could do of perhaps identifying older cells that need to be replaced sooner rather than later, and then even maybe removing them, or leading to their elimination. That seems possible to me. It's not my field, but that seems like it would be possible and potentially helpful. And maybe could even be part of this silencing in some way. And then in terms of replacing, having nanobots, I guess they could guide cells to places where they need as well. But again, it's not my field. I'm just imagining that this is in theory possible. And I'm not up enough on the field to know when this might ever occur, but yeah, sure. I'm always surprised at what other fields are accomplishing as well, including nanotechnology. So who knows?
Mm-hmm. Yes, and that's one of the fascinating aspects of living in our era. All of these different areas of technology and research are developing in parallel. We discussed the race between artificial prosthetics and lab-grown organs, for instance, and it would be fascinating to see what nanotechnology has to bring in terms of enabling these kinds of synergies among medical approaches. Now we have a question from Jason Geringer, one of our US Transhumanist Party members. So Jason, please feel free to ask it.
I have heard that I'm not sure if this is true, but that there's this theory that the reason for psychopaths, narcissists, sociopaths, is that they either have a slow amygdala, or not a slow, a small amygdala, or an amygdala that isn't connected very well to the rest of the brain. And if that's true, then is there a possibility we could use your grafting procedure to graft onto a small amygdala and possibly give that person more empathy?
Yeah. What you're bringing up in a way is this broader question of can we not only make things younger but make things better, right? And so I'm not familiar with these studies that have looked at the amygdala in these individuals, but it's certainly possible that the amygdala is involved in this. And so, I mean, the answer is yes. I mean, you could, right? If you knew how it was different in these individuals, you could take steps in these replacements to try and make things better. You know, that's another area that, you know, we're not addressing. We're not, you know, we just want to make things younger and healthier. But you know, because when we start talking about making the brain better, more functional, first of all, I don't know that we know enough about how to do that. I think that it might be a bit premature to be thinking about that. But then, you know, then there's a lot of other social and ethical implications that would need to be discussed. But yeah, I mean, there's no reason not, right? Even at the, I mean, it's certainly something that would be easier and easier to convince people of is, for example, if you did have a degenerative condition, right? So some of these have genetic components to them, and we know what they are. And we have the capacity now with, I don't know if you've heard of CRISPR technology, but to change those genes to what is the normal functioning gene. And so since we're starting from cells and culture, we can do this. This is something we can do now. So when we put when we repair the tissue, in that sense, we can make it better. And I think that's an easy sell, right? I mean, when it's a clear disease. When it's a mental or a psychiatric disorder, things are generally much more complicated. The know-how to do that is not there yet. But you know, maybe if there are clear indications of how to make things better, you know, one day I think that will become a very pertinent question.
Yeah, I mean, how awesome would that be if we could cure psychopathy? That'd be a good thing.
I mean, these things tend to be very complicated, though. You know, all these broad syndromes, schizophrenia, we don't know what they are biologically yet.
Can you graft right onto a healthy young brain?
Yeah, I mean, everything works better the younger you are.
Mm-hmm.
Grafts would do better. You know, you recover more from surgery more quickly. Yeah.
So I wonder if there was like a part of the brain that made you a better piano player, you know, savant. Could you like get some boosts in that, you know?
Yeah. Well, I mean, that's the idea with like stroke, but I guess you could do it even if you don't have stroke. But you know, stroke, most of the disabling long-term effects are with speech and motor function, right? Those are the ones that people care about most. And so that tissue is gone if the stroke affected the language center. But putting in new tissue the level of plasticity of a new tissue is much greater than an adult tissue. And so the expectation is that you would be able to relearn language more like a 2-year-old than an adult, because you have this blank slate that you're trying to use to learn language. So it should facilitate things greatly. And I don't know where the center is for piano playing. It's probably somewhere in the auditory center, but likewise, that there might be an opportunity there as well.
Yes, I think that kind of cognitive enhancement would be astonishing for the progress of the human species, and of course for the quality of life of the individuals experiencing it. And Jason brought up an extremely important aspiration of transhumanism, which is to use cognitive enhancement to fix some of the mental issues that unfortunately afflict certain individuals. And I've come to the realization over the years that the preponderance of these issues in contemporary societies is high enough to be a serious detriment to progress. If there were a way to effectively resolve them medically, we would be in a much better place as a species, including facing many fewer existential risks. But as you pointed out, this is an extremely complex and challenging area. And I think we are likelier to get to indefinite life extension before we get to effective reversal of these kinds of complex mental issues. So our challenge as transhumanists in the meantime is essentially to figure out ways to muddle through, so to speak, by means of having good rational institutions and relying on the rational capabilities of people who don't have these issues to avert any of the problems that may arise. So we're definitely in for an interesting few decades, to say the least. Now, we have a question from Didier Cornell. Once again, he's interested in knowing what is the best place online to get more information on your project, because he has observed that a lot of these ideas seem to be in fairly early stages. So he wants to know where he can go to learn more.
Yeah, I mean, I wrote a review a couple of years ago with Jan Vij, but again, a couple of years ago, we made a lot of progress in how we think our approach is going to work better since then. So I think the more recent thing is actually my book. I mean, I hate to, shamelessly promote my book, but I think that has, a third of the book is on how we would achieve a brain rejuvenation. But even my book, which, I wrote a year ago and it's been out since the fall, our thinking and our enthusiasm for why this is going to work has changed, improved, because we've thought of a lot more ways. There's always caveats coming up, but we're finding ways in which, okay, we can work around that. We can still get it to work. So our confidence has built, and along with our proposed methodology. So, I mean, none of this is published out there anywhere. If you're interested, just send me an email, and we can chat in more detail about this. That would be the best way to do it, short of buying my book.
Excellent. So I did link to the Amazon page for Replacing Aging within our YouTube chat, and also I will post your email address there for our viewers who are interested in contacting you for more information. Now I would like to go to Art Ramon Garcia. I know he had some questions for you as well.
Yes, I know they have hippocampal artificial replacements, and I guess it's like a silicon chip. Is there anything you can leverage from these hippocampal prosthetics into your tissue replacement? Like, can you do it simultaneously and then put them in different parts of the brain and get them to communicate? Any idea, any thoughts?
So I don't think they have quite achieved a functional hippocampal replacement yet. Um, there, there is an effort to do so. Again, competing efforts with biological replacement as well as a silicon-based one. And the hippocampus, yeah, I mean that's an area where there is a maybe more of an opportunity to replace at least unilaterally at first until it integrates, and then the contralateral side subsequently, but replace the whole structure at once, because although the hippocampus is essential for what we do every day, right? It does many things, but maybe the best known and maybe the one that might be most important to us is to form new memories, which we kind of take for granted, but we need our hippocampus for that. So I think that you would want to do at least partial hippocampal replacements at the same time as you're doing cortical replacements, especially entorhinal replacements, which are closely connected with the hippocampus. So you definitely want to coordinate that. I don't know if I'm answering your question or not, Bill. Were you asking more in terms of sort of using the same replacement technology?
Well, I hear that it's a working technology. I mean, I'm reading an article from 2003 where world's first brain prosthesis revealed. So it's been a while since they've had these hippocampal artificial replacement chips. So I thought maybe since it's been so long, there's something you could leverage from that type of research.
Yeah, I mean, maybe you can send me that link, but I really doubt that it's a true replacement. It may be a prosthetic that might help form new memories. Is that it there? In the chat?
Yeah, that's one of the articles I found. And there are several others. There's even a Wikipedia article. So I remember reading about this with wounded vet programs. I think DARPA had a program for wounded vets. So, and I think it's a rather mature research.
Well, at least in this article, right, they're just, starting, proposing to test it.
Yeah.
So I don't
But it's
know
2003.
I think it's only in animals right now. I have not seen it in humans personally, and I believe it also may require like an external interface. So it's not like it requires a computer connection essentially, which adds a whole nother level of complication and
like most parts of the brain.
Yeah.
I think what Dan is saying is correct. Like most parts of the brain, the hippocampus has long projections to other parts of the brain, and unless these chips can recapitulate those long connections and also adopt, biological connections from those parts of the brain, it can be an aid, like maybe like a cochlear implant, amplify signal, or but I don't see how something like that could act as a replacement. Not yet, anyway. Not in that current form.
Well, while we're on the subject of the hippocampus, there's been a lot of debate about whether there is neurogenesis taking place in the adult human hippocampus. There's lots of research showing it occurs in other primates, but there's a lot of controversy about whether it occurs in humans and to what degree that is significant. So I was curious because I read that theoretically what might be happening is that new neurons are born in the hippocampus and then they sort of migrate out. So I was curious if you have any, if you've reached any conclusions about that, and if that's if the decline in that is an important part of aging, and if that could somehow be reversed through either pharmaceuticals or some other intervention?
Yeah, I think, if neurogenesis does go on in the adult human hippocampus, for decades, and there, like you say, there's still some controversy over whether it does or not. I think enhancing it should be possible, either, even pharmacologically, because we know, what fair amount about that process already, and we have lots of examples in animal models of how to increase it as well as decrease it, but in this case, we'd be more interested in increasing it. So we know what some of those pathways are. So they're fairly, activation of a simple pathway might be sufficient to increase neurogenesis. And would that be beneficial? Probably. It would probably have some.
Yeah.
Yeah.
But it seems so. There's a lot of like alternative medicine people who are promoting certain mushrooms, like lion's mane mushroom, and all different things, saying that they boost the production of new neurons. But as far as I can tell, there's not really much good science behind that. And also, it seems like the number of neurons is very, very small. So I read like they've observed, new neurons being produced in one study at a rate of like a dozen per day or something. And to me, like that doesn't seem very significant at all when you have 100 billion neurons, right? So it's curious, yeah. So yeah, I think it seems like the neurogenesis might be somehow, helping with plasticity. But it seems, I think a lot of the claims about that have been exaggerated, basically. That's my impression.
Yeah, I wouldn't disagree with that. But what's important to keep in mind too is that these stem cells and this neurogenesis is very restricted in space, right? The migration that occurs is just microns.
Oh, really? Really? I didn't realize that. I think I had been misled by kind of bad journalism.
Well, there's, at least in the rodent brain, although this I think has been shown not to occur in humans, there's another area of neurogenesis that goes to the olfactory bone. And those will migrate greater distances. So, at least maybe 3 millimeters in the mouse. So that's a lot further. But that neurogenesis seems to die out before you reach 2 years old in humans. That seems less controversial. But in the hippocampus, and there's an area of the hippocampus called the dentate gyrus, that's where the neurogenesis, it's controversial whether it's still going on in our brains. But if it is, it's very restricted to that area. And the stem cells, once they become postmitotic and migrating neurons, will move, 5 or 6 cell bodies in diameter away from where they were born. So it's a very short distance. And they all reside in this dense cell layer called the granule layer. And they're very specialized, right? It's not like these cells can give rise to neocortical neurons, right? And they don't migrate there, and they don't have the innate capacity to give rise to neocortical neurons. So they're very specialized in what they give rise to. There may be some in the hypothalamus as well that have been described. But again, these are very specialized and restricted, not only in where they go, but what they become. So there may be some opportunity there to rejuvenate function by stimulating those, but it's restricted to very small, limited parts of the brain, if it occurs at all.
Interesting. Yeah, I think that's been very much overhyped in the press, unfortunately, and now it's starting to come out that it's been overhyped.
Yeah.
So our next question comes from Danielle Marsden, who is interested in how the price of these procedures in the future could be lowered to make them more affordable. Because, of course, right now, going for brain surgery is one of the most expensive procedures that a patient can undertake. So let's say this becomes a commonly accepted method of reversing biological aging, and it becomes fairly expected for people, say in their 70s, to go have this procedure while they still have a lot of fairly healthy aspects of their brain function. So how would you be able to perhaps scale it or make it sufficiently widespread that you could take advantage of economies of scale? Would you have to rely on a lot of automation to make that happen?
I mean, that's a good point. I mean, automation, like I alluded to earlier in terms of the surgical techniques, would greatly accelerate the implementation of these grafts, but also the costs probably would significantly decrease. But I mean, you know, and I'm not the one, again, that's an expert on this. Aubrey and others have thought much more deeply about the cost benefits of doing this and how it can be, you know, affordable and accessible to everyone as they get older. And you know, we know what the costs of getting old and being unable to work are, right? It's a huge cost on society, the healthcare costs of that. And that goes on for decades, if you're lucky enough to live for decades being old and retired. So, you know, the argument's been made, and again, I'm not an economist, I haven't crunched the numbers, nor will I will let others do that. But the argument's been made, and it made sense to me, though, that you actually save money, right, overall, in getting a person in a state where they're young and able to continue working instead of draining all this money to take care of their health care without paying taxes. And so, you know, assuming that's the case, then the goal is to just, you know, get the leadership that sees this, and of course, you know, it's all about convincing enough people to vote for that leadership, to take the, you know, to make this happen, right? So I mean, it should be something covered by healthcare for everyone, just like healthcare, everybody should be covered by healthcare now. And so we're already somewhat at a deficit because not everybody is. But those treatments should simply be incorporated into healthcare and be covered because they will be good for society as a whole, both economically and in terms of you know, standard of living and health.
Aubrey de Grey has stated in his conversation with us that he foresees that there will be an inflection point in terms of public attitudes toward the reversal of biological aging when large numbers of people start to see that it is feasible. And he believes this inflection point will happen before the actual technological feasibility is achieved, but rather when that feasibility is in sight for a lot of individuals. And you're correct, that will be the point at which we will need to have effective methods of political advocacy in order to get lawmakers to recognize the importance of making these treatments available to everyone as affordably as possible. Indeed, this is why the Transhumanist Party exists. I would say our most important goal is to prepare for that inflection point so that when it happens, we are ready to go at scale with this kind of advocacy. So these are great conversations to be having. Didier Kernel has one last question for you. He asks, do you have any promising contacts with people from the Human Brain Project of the European Union, as well as the US Brain Initiative and Brain/Minds from Japan?
Yeah, not so much with the foreign countries, although we've collaborated with labs from around the world previously on different projects. So we're, there are no borders for science and progress and knowledge and technology. But in terms of the Brain Initiative, I mean, there are groups here that we collaborate with that have gotten funding for that, I believe, or certainly they've applied for it. It's a huge initiative, right? So they give a lot of grants out to a lot of researchers at different institutions. So, and some of what they do is part of how we're analyzing our graphs. So they have ever better brain imaging technologies that we're taking advantage of as part of collaborations. Whether those particular labs have received Brain Initiative funding, I don't know, but they're certainly benefiting from the technology, and we're certainly collaborating with them. So, yeah.
Excellent. And then we have another audience question from Jennifer Hughes, who suggests that the approach of repairing damaged cells may compete with replacement. So where do you think the obstacles are to repairing damaged cells? On the one hand, we do have aspects of the SENS approach that focus on that, for instance, clearing out junk from within cells or repairing mutations to nuclear DNA or mitochondrial DNA. Do you still think that's more challenging than just grafting on healthy younger tissue?
I do. I think it's doable to replace and highly questionable whether you can actually repair. I mean, you just look back over the decades, right? We've had the telomeres. Oh, if we fix those, maybe we can reverse aging. Inflammation, DNA mutations. I mean, there's just a long laundry list of things, of targets of repair that have been created excitement for a while. And then people just add on more and more targets over time. And we know from biochemists from decades ago that there's hundreds or thousands of forms of damage. So I think repair is very ambitious, to say the least. I think replacement is much more likely to work in the short term. Yeah.
So we just got another question from Brad E, who asks for your thoughts on in situ regeneration of neurons in the neocortex with epigenetic resetting via transient or partial reprogramming factors.
Yeah, so the partial reprogramming, setting back the maturity state of neurons, it would be very difficult to implement, right? I mean, reaching the neurons specifically to reset them, or I guess maybe you want to reset all cell types, but some of them are already in a proliferative state, or at least capable of proliferation, like astrocytes, oligodendrocytes, microglia. It gets very difficult to think of ways in which just by resetting the epigenetics back to a sort of an early developmental stage is going to work. I haven't heard anybody come up with a way that can be implemented. But even if it could, right? Maybe it could. But then it still doesn't deal with the damage that exists. in all the extracellular space of these neurons. So that's not going to go away just because you sort of reset an epigenetic clock. And these cells, some might argue, well, the cells, the young cells will produce new extracellular matrix. Not if they're already embedded in existing extracellular matrix. There's they don't produce excess. They get feedback that there's already extracellular matrix there. It happens to be all damaged and old, but they're not going to replace that on their own. So you're still going to have an old brain. So in that sense, this resetting of the epigenetics, I think it's going to be very limited in application. But that's not the only use of epigenetics. So I think there's other uses of epigenetics that can be very fruitful. And we saw one example from David Sinclair's lab, I think in the last year, with the regrowing of the optic nerve, for example. So by putting cells that are in a mature state in an immature state, so they now behave like young neurons, you can get some regeneration, right? So I think but that sort of comes back to the axolotl thing, right? I mean, it's another form of replacement, and I think epigenetics can be effective there. Maybe even to grow a new limb at some point. It would certainly be a part of that process, but it's not in terms of fixing what's there without replacing with new projections or new limb or new cells. Just changing the epigenetic landscape of a cell is, first of all, not easy to implement in a way that would be effective. If you start thinking of all the different cell types, the different stages of maturity, it gets really complicated really quickly. Not to mention that you'll be flooding the system with a delivery system that is probably going to cause more damage than do good, like if it's viruses that you're using to deliver. So the implementation is a problem. But then also the benefits would be very limited. So in that sense, I don't think so. But in the sense of regrowing things, I think epigenetics can play an important role.
Interesting, thank you. And now we have a question from Jason Geringer. Jason, would you like to pose that question?
Sure. How much do you think we could add to a young, healthy brain? I mean, is it possible, do you think, in the future, if all goes well, would we be able to get like a skull extension and like double, triple the size of our brain? I think that would be possible. Or even, would we even want to?
Yeah, I mean, it wouldn't be impossible. I mean, there's no physical reason why that we wouldn't be able to do that. But yeah, the question would come down to is more better, because what's really important is the wiring, right? So you'd have to ensure that those additions are wired in a way that is useful to the functioning of the whole brain, right? So not impossible, but we wouldn't know how to do that now. But yeah, in the future, it's not impossible. Yeah.
So I'm curious with regard to your thoughts on this in terms of brain augmentation. How easy or difficult would it be to graft an electronic component onto the brain that would interface with the brain, either an artificial neuron or perhaps even like a computer component that would have data storage or advanced computation functionalities? How far away are we from that kind of integration?
I mean, it's already happening, right, at some level. And there's examples from years ago where these brain-computer interfaces have allowed quadriplegics to move their limbs again, not just move their limbs, but grab a cup and drink with and coupled to these deep learning algorithms. These interfaces allow the computer to learn very quickly what you want to do and execute it very, in a way that you can actually learn to drink from a cup fairly quickly. And also not just normal limbs that were implanted with electrodes to control the muscles, but also artificial limbs. The same thing was done with computer-brain interfaces for movement. And they've been applied for things other than movement as well, like voice boxes, and certainly in development. So yeah, it's already started. But that's sort of more one-way street, right? So those brain-computer interfaces are reading our brains and executing functions or tasks. The other way, there's also examples of the other way. I just don't know if they've been used therapeutically in humans, where you can stimulate certain parts of the brain and activate. Certainly when surgeons are doing brain surgery and they poke around to find out where your functions are to avoid important areas, you're awake and they can map what's evoked in your mind when they're stimulating certain areas. So the other way is also possible. You can certainly activate parts of the brain with electrodes, and in principle those can be hooked up to a computer. So you can get two-way brain interfaces operating that way. And so that's just going to grow. I mean, that's just going to get better and better, right? Although the way, I don't know if I should go there, but maybe you guys are the perfect crowd for this. I don't know. But
Go for it.
Yeah, the way people talk about, a brain download or mind upload, the way it's currently described doesn't make much sense to me, because they're really just making copies. And you can erase that copy and it won't affect your consciousness or who you are. Or you can make 100 of those copies and activate all of them and then, destroy them all, and that won't affect who you're, there's separate. It's a copy. It's not the same. But there are ways which are being developed now where these brain-computer interfaces can get better and better. And so one wonders, at what point, if the AI in the computer is, and it's a two-way street, is communicating well enough with your brain, at what point is there sort of a melding of the two, right? I mean, where if you then shut off the computer, you feel like you're less conscious, like there's things you can't think about anymore because your synthetic side was doing that for you. So it's possible that these technologies get good enough, or I don't know when, but at some point in the future to accommodate this. But I mean, we're certainly not there now. The fine electrodes they're getting better and better. I mean, Neuralink is an example of this, right? Where they have these very fine carbon electrodes. I think that's a great improvement over. It's not the only. They're not the only ones who are developing these very fine, sort of non-inflammatory or minimally invasive electrodes. They're probably the more visible ones, but there are others as well. So the technology is getting better. But yeah, I don't know. Sometime in the future, there might be a possibility to become more synthetic. Certainly, if we accept that that plasticity can occur from one biological substrate to another biological substrate, there's no logical reason why you wouldn't accept that that the transfer could occur to a non-biological substrate, assuming it could hold function in a way that we recognize as part of our minds. So I don't know.
Yes, I actually quite agree with your observations. I'm not convinced that many of the, let's say, cruder or more dramatic approaches to mind uploading would actually preserve the identity of the individual for the very reasons that you mentioned, that they would create just copies and there wouldn't be this experiential continuity. But if one could obtain a very thorough integration between the biological and synthetic components of the brain, so that the synthetic components start bearing a lot of the cognitive load, then perhaps we could have partially synthetic brains in the future, or even at some point, hundreds of years hence, wholly synthetic brains if we continue that procedure to replace the biological components with the synthetic components. I think we would have to be very careful to make sure that the synthetic components preserve the ability to experience the world directly the way that biological neurons do. So that would definitely be a fascinating area to consider. What are those prerequisites of direct experience so that these components, they don't just process data, we feel through them.
Yeah, no, absolutely. It's a lot of fun to think about at a lot of levels, philosophically, you know, and as well as technologically.
You know, one thing I wonder if we can actually get, you know, that going with synthetic and the connection time, and we start thinking faster. I wonder if that would like completely change our perception of time itself. Have you ever heard of like people, oh, my life flashed before my eyes? And I think it's like this phenomena that they were like just thinking really fast at that point, you know, because they're about to die. And I wonder if, do you think thinking faster like a computer would change our perception of time?
Possible. I don't know how we perceive time now. I think it has something to do with the cerebellum, but I'm not sure. The cerebellum certainly has to do with timing of movements and coordination, and maybe also perception of time, but yeah.
So we have some follow-up questions from Brad E. His first follow-up is to express surprise that the extracellular matrix isn't actively maintained by what genes are expressed. So I'm curious if you could elaborate on that.
Oh, yeah. So, I mean, in some parts of our body, it doesn't turn over for a whole life, right? So in other parts, it might turn over a little faster, and it depends where. You know, the skin is a good example. So the superficial layers get turned over very quickly. In fact, they're not very cellular to start with. But the deeper you go, you know, the longer things stay around. That's why tattoos, you know, don't go away in part. And so, you know, it depends what part of the body. But the brain, you know, damage starts accumulating in the extracellular matrix at a frighteningly young age, although there's no, you know, cognitive deficits associated with that, thankfully. But it's very progressive. I don't know if I could say it's linear, but it does happen early and it just progresses throughout life where you get more and more damage. So that, you know, the cells aren't replacing those proteins and that damage and those aggregates and those covalent links. There's nothing there undoing those covalent modifications, and so damage just slowly accumulates. And if you put in new cells in these old matrices, you know, the matrices remain old. They don't get replaced. I mean, that hasn't been shown in the brain, but in other parts of the body, even like the hematopoietic system. If you put young hematopoietic cells into an old bone marrow niche, they behave more like old stem cells, for example. You know, I think that that's a big problem. Is not just the cells, but you know where the cells live, and that greatly impacts their ability to do their function and eventually to survive. And especially for neurons, you have all these long projections. If they're in an environment that is not allowing them to do their job, they'll degenerate, and that's essentially what happens. So you could set their epigenetic state back to an immature state, but I think that would have very limited benefits.
Now, if I recall correctly, it used to be thought that new neurons do not form at all during the lifetime of an individual, but recently there has been evidence of some new neuron formation. It's just not enough to overcome the general tendency toward decay. So how frequent, how prevalent is the formation of new neurons? Is there anything that people can do to affect that, short of getting a graft, or are we pretty much fighting a heavily losing battle with the technologies we have today?
Yeah, I mean, that's sort of, we touched on that a little bit with Dan's questions earlier. And so, yeah, there's pretty good evidence that there are no new neurons in most parts of the brain, including the neocortex, over a lifetime. The neurons that are there now were there when I was a teenager. The hippocampus is an area that's in size very small compared to the neocortex, and only part of the hippocampus is there potentially new neurons that are generated in humans. But this is still somewhat controversial. I mean, it's people. It's not clear and just how much there is and whether it's significant is not clear either. So you could augment that, and it might help. You could try to augment the amount of new neurons that are being born, and that might help that function in the hippocampus. But it would, it's not going to help rejuvenate the rest of your brain.
Yes, indeed. So we definitely need some advancements that facilitate that. And it seems that bringing in new neurons from the outside, which is your approach, is, in your view, the easiest and most straightforward way to do that. So we have the second follow-up from Brad, who asks, do you know of any open calls for research PhDs on neural regeneration with lab leaderships that share in your philosophy on aging?
So there isn't much crosstalk between regenerative medicine and aging, which is a big problem. So maybe a little bit more and more. There are other labs doing transplants, and they probably thinking about aging, even though they're not talking as much about it as I am. There's a list of labs. I mean, again, if Brad wants to contact me by email, I can give him specific examples of who might be worth contacting. Yeah, I mean,
yes, indeed. Well, hopefully Brad will follow up with you. He seems to be quite interested in this research and perhaps getting involved with it. So now we have come to the point in our salon where we will go around to each of the panelists and see what final comments or questions they may have. And then, as is our custom, we will let our guest have the last word. So let us start with Art Ramon Garcia. Art Ramon, please proceed.
Yeah, Jason touched on it earlier, but preemptively seeding the brain with new tissue that's been specifically upgraded genetically engineered, maybe cells that don't die, that would eventually replace the natural cells that will die. Um, Anything like that, to maybe sell that to someone who has the money, sort of like selling a hair transplant before you go bald, you know. Any idea?
Um, yeah, I mean, I agree with you that the earlier you do this, the better, right? The earlier, at least you start this process. But in terms of seeding the brain with cells, you could potentially do that as well. But there'll still be an accumulation of damage, right? And again, it's not just the cells, it's their environment. So even if you're replacing the cells, I still think you need a way of removing all the damage that accumulates outside the cells, as well as, if you replace the cells, obviously you'll be replacing the damage that's inside the cells. I think, that there's definitely room for a complementary approach there, where you could have a seeding of cells that could act as stem cells. And that's also where, an epigenetic approach can contribute, and people are doing in vivo reprogramming of certain cell types to neurons. So that's an epigenetic trick where you can convert one cell type to another. They're looking at it more in terms of in response to injury. Because there are no new neurons born, maybe you can get these cells to produce new neurons. And that's had some success, but the idea, I think, could work. But in terms of, it's a little harder to think of the cells just sitting there over decades and being active when you want them to replace, specifically the cells that are going downhill. I think that might be a little harder to do.
Okay, thank you for coming.
Thanks.
All right, now let us go to David Shoemaker.
Yeah, thank you so much for being here today. Enjoyed this. Matter of fact, I enjoy it enough to go buy your book. And then the follow-on question is, as your work continues to progress, where can we that are interested in this follow this? Will you be published in various journals, or what are your plans?
Yeah, I mean, we're going to keep publishing as long as we're in academia. Once we move to industry, that's not necessary, but I'm sure you've heard of the publish or perish. We don't really have a choice.
Right.
So yeah, this will get published. Some of our work is, something that I didn't tell you about is being patented. And I did not tell you because it's a secret or anything, but just because it's not directly related to the tissue engineering project. So, we are looking into to what we can patent for the project in terms of tissue engineering, so that that might be might require holding on before publishing a little bit, but I tend to be very open about this. What we do, I tend to tell everything to everyone, because I think that's how we can make the quickest progress and share things. I'm a little more tentative now because I'm getting a foot in the business side of things, and funding is sort of closely tied to intellectual property and being able to maintain that. And so I'm a little more careful about that. But we will publish everything at some point. And if you call me, I'll tell you everything I know.
Thank you very much.
All right, thank you. And now let us go to Dan Elton.
Yeah, I don't have much to say at this point. I think this work you're doing is extremely valuable, and I just wish I hope that there will be more funding coming through for this type of work from the National Institutes of Aging and other research bodies. And if there's anything I can do to help promote it, I'll be there. So,
Great.
thank you.
Yeah, you're there at the NIH. Yeah. So,
Well, yeah, I don't think I could help in that regard, but I'd like to, but yeah, a different building, but yeah.
Yeah. And the interest has come more from NINDS, and I think will continue to, rather than NIA. I wish the NIA.
That's interesting. Yeah.
More, but yeah. So yeah. But thanks. Yeah.
All right, and now let us go to Jason Geringer.
No more questions, but I'm glad you came here, and this is like awesome news. I mean, it's always good when you grow up thinking that something's impossible, and then it's like, oh yeah, we can do that now. Gotta love that. And well, thanks for coming.
Thanks. We're not there yet, but to me, it's a question of time. It's something that's achievable, and we will achieve it.
Yeah, it's like
we don't have anybody walking on the surface of Mars, but we know it's possible.
Right.
I think that's where we are.
Yes, indeed. Well, every great idea was at one point just a concept that somebody proposed, and of course someone has to achieve it the first time. So I'm quite hopeful for the next several decades of human history that we do have breakthroughs in all of these areas, from colonizing Mars to being able to replace components of the brain to being able to repair cellular damage. I know you think that's more ambitious, but I also think that a lot of promise is found within the other strands of the SENS program. And also, I want to point out, I know it's late in Spain, so I don't know if our friend Jose Cordero is still with us, but he definitely quite enjoyed your book, and I think there he is. So I want to offer him the opportunity to ask any final questions or make any final comments.
Well, again, congratulations. I truly admire what you are doing, and I wish more people keep on following this replacement theory. I have a question. I don't know if you are aware of a Middle East foundation that is supposed to be funding a lot of money in the order of about $1 billion per year in terms of anti-aging rejuvenation. Are you aware of any of that?
No, but I would greatly appreciate you sending any information or contact information, even better, if you know somebody there.
Yeah, actually, it's
best with introductions, but if you know anybody.
Actually, it is a secret still now, but Nir Barzilai is supposed to be scientific advisor.
Okay, that's the secret. Yeah, I've heard Nir talk about this secret, but I didn't know it was connected to this.
Yeah, he announced it in November for December, and then again last month for May. Let's see if it comes out in June or July.
Yeah. So I need to talk to Nir about this. Yeah. Great. Thanks.
It's interesting. All sorts of secrets may be revealed in time. So I look forward to the time when these secrets can be more openly discussed. Perhaps we'll have some follow-up salons that incorporate conversations on some of these ideas. So now
one more point, because Jean speaks French. I just published a book in French that you can see behind me, La mort de la mort.
Ah, yeah. Well, that's your book. Yeah, yeah.
Yes,
You had it translated. Excellent. So
yes,
I can reread it in French now. Excellent.
that's right.
Thank you.
Wonderful. So now we've come to the point in our salon where we will let our guest, Jean, offer any concluding remarks about this conversation or anything else that you think needs to be said on the subject. Please proceed.
Yeah, no, big thank you is basically what I want to say. Thank you a lot for having me. And also thank you for what you're doing. I think, progressive leadership we've seen as not always present. And so we need as much of that as possible, and more as we move forward. We need leadership that will responsibly incorporate technological progress into making our lives better. And so what you're doing, I think, is important now, and it's just going to get more and more important. And I look forward to you guys playing a bigger and bigger role in this. So yeah, thank you very much.
Yes, indeed. Thank you. And that is the trajectory we hope to set ourselves on in the coming years. Conversations like this one are crucial to making that happen. To everyone watching this virtual Enlightenment Salon, these are the kinds of conversations that we believe need to shape the future of politics and of the human species, to have our future directions be informed by science and innovation and by the imperative to apply their fruits to the benefit of as many people as possible. So thank you, Jean, for participating today. We learned a lot from your presentation and your answers, and thank you to our audience. We have some exciting virtual Enlightenment salons lined up in the coming weeks. Next Sunday, on April 18th at 1 p.m. Pacific time, we will have as our guest Andres Gomez Emelson. We will have a fascinating discussion on qualia and consciousness and some of the research he has done into those areas. And then on April 25th, the following Sunday, we will have as our guest the renowned Dr. Mary Ruart, who will discuss some of the obstacles to research progress at the Food and Drug Administration, following up on our panel discussion on FDA reform and what can be done to alleviate some of those obstacles. So we have a lot to look forward to. And in the meantime, live long and prosper.
Okay. Thanks again, Gene. Yep.
Thank you, guys. It's been a pleasure. Bye-bye.